Good afternoon. Hopefully everyone will stay awake since you just had lunch. I will make one small clarification. I'm one of those rare beasts that's a staff scientist, but not a PhD nor MD. I'm just lots and lots and lots and lots of experience. Okay, so thank you very much to Global Engage for inviting me to speak. Thank you all for showing up here today. My world really is more focused in spatial proteomics. And we have three major platforms at Fred Hutch. We have the kind of MIHC multiplex up to 8 markers. That's the Opal or PhenoImager or whatever name you want to call it. And that is in our experimental histopathology research core. And then we have the Luna-4 Comet. That's going to be a bit more mid-range. 10 to 15 markers is where it really shines. You can, of course, do more, but that's where we use it. And that is in our 𝐒𝐤𝐞𝐚 histology lab, the clinical trials pathology lab. And then my core, the translational pathology, does the phenocycler, and most of our panels are about 45 markers. So as these proteomics have expanded and advanced, and we're doing more and more and more markers, spatial transcriptomics has, of course, also been taking off. And I get asked all the time, why am I still focused so much on proteomics when you can get so much more data out of transcriptomics? And the long and short of it is, it's a little complicated. So I'm going to tackle that, talk about kind of merging of the two, how we're developing out some of the data hurdles that we've had, and looking ahead. So what we're really talking about when we're talking about spatial proteomics versus transcriptomics is mapping protein expression versus mapping gene expression. These are different beasts. On the transcriptomic side, you're talking about hundreds or thousands of genes, and you are measuring the abundance of transcripts. It's pretty distinct. It's fairly straightforward. But inherently, RNA is unstable. It's going to fall apart. Biologically, it needs to work that way. But it also means that now you need to be working with some fresher samples. It's also costly. It's going to be even costlier than some of these really high-plex proteomic assays. And in this day and age, budgets have gotten really, really tight. And so it's even harder for investigators to be able to afford this. On the proteomics side, for one thing, it's very visual. I'm very visual, so the fact that I can see the macrophage eating that tumor, it's awesome, in my personal opinion, but it's also used diagnostically. These are your drug targets are going to be the proteins, and proteins are more stable. So you can use those archive samples, the archive slides. We work with FFPE. In our phenocycler, you could technically use fresh frozen. We don't use that method. I'm trying to avoid it like the plague because it's just, for me, pretty means analyzable, and fresh frozen just doesn't have quite the morphology. It's less analyzable. And so for me, that means less pretty. I know everybody has their own opinion on that, but proteins are messy, right? So those biostatisticians, the bioinformatics people who really like working with spatial transcriptomics data, it's fairly straightforward, really hate, for the most part, working with protein data because it is not straightforward. Now, some of this has already been tackled a little bit for this morning, but, rehash because we're all talking about the same thing. But here's one of the big things is that when it comes to proteins, you have a limited number of antibodies. When you're talking about genes, you can sequence the gene. You can make a probe that's going to target the gene. When you're talking about proteins, that gene the RNA has gone through post-transcriptional modifications. You also have these feedback loops. You have other things that are going on so that the proteins at the end of the journey, you may have variations associated with that one gene, or you may have no protein at all. Maybe you have those transcripts there and there's no protein that is expressed. So the long and short of it is your RNA abundance does not necessarily equal protein expression. Oftentimes those kind of bigger population, the T cell, CD3, the macrophage, those tend to be more correlation, more stable. But when you get into the functional markers in particular, that's where you see more of a difference. And so combining these two, the transcriptomics and the proteomics, gives you that more robust story. And people had already been tackling that. They were starting to see it even before the spatial world, when you're looking at just single cell and seeing some of these differences. And then people started to bring that into the spatial world, looking mostly at serial sections, but still you are seeing this disparity. And now it looks like it may actually be very informative, right? In many cancer types, the more correlation you have between your transcripts and your proteins, the worse the outcomes, which makes sense because in cancer, you are disrupting the regulatory processes that were creating the differences to begin with. So the JAX lab put up this protocol, it's on Protocol.io, where they have taken slide through Xenium, then through PhenoCycler, and then H&E. So I just happen to really love the graphic. It's going through the transcripts and the proteins, but the whole point is you're doing it all on the same slide. Then you're going to be able to register them and move your analysis forward, which I will point out is not as straightforward as it sounds. So I roped in the FILD, the Fred Hutch Innovation Lab, and they're the lab that is going to bring in new technologies. It's going to tweak it, update it, get it ready for Fred Hutch, and then, put it into motion. And they have the Xenium. And so I said, I would really love if we could collaborate so that you guys run the Xenium, I'll run the PhenoCycler, and everything will be great. Now the problem was, how do you get investigators to offload that really expensive run for the Xenium for you to do PhenoCycler? And then before the H&E is done, which a lot of people use the H&E after the Xenium, and we hadn't even figured out the protocol to get the flow cell off and verify that we could do a good H&E when this all first started. We can now, by the way. So we have this process that we're looking for volunteers, and our first volunteer, Dr. McPherson, unfortunately is working with mouse. All of our stuff is human. So we had some antibodies that were reported across to mouse, and he knew that we were going to throw this on. It's not optimized. We'll hope for the best, but this is all proof of concept at this point. And one of the first lessons is size matters. And then the next slide is going to be the one that everybody takes pictures of. Okay, so we have the PhenoCycler and the Xenium. Where you see the blue, that's the acquisition area. Your tissue has to fit within that space. And to give you perspective, over here we have some, which I'm sure y'all can figure out over in the corner. We have a TMA. This size fits within the 15 by 15 millimeter acquisition area of the original codex, which was all done on cover slips. So we have all these investigators that set everything up for the codex. And now that we have our PhenoCycler, next generation, here's your secret. The PhenoCycler is the codex, just renamed, and add the fusion. That's just a different microscope. So now we can fit two of these TMAs on the same slide. Happiness and joy abounds. But on the Xenium, you can already tell you can only fit one TMA, and you also have to chop off some cores. For giggles, we're going to throw in the Comet and the Cosmic. So those are the other two high-resolution proteomic and transcriptomic platforms that we have. Like I said, the CTP lab has the Comet. They have the first generation, so that's just a 9 by 9 millimeter. But our experimental histopathology core put in the request, please let us have the next generation, which has 12.5 by 12.5, but budgets, nope, not this year. All right, so Cosmics is able to do transcripts and proteomics. You see that it at least would be able to fit one whole TMA. I'm not going to discuss the pros and cons of all the different platforms, but that one is not for us. And I'm going to go ahead and throw in the Visium and Visium HD. Those are whole transcriptome, but still you get all this extra data. The Visium is low resolution. You're looking at cells, whereas the Visium, you get down to the cellular level, but either way, you're looking at really small space. So getting back to our projects, that first mouse one, we learned a lot. And the most important part is we were very encouraged by what we saw. The second one, we had human bone marrow. Human, yay, more markers. Bone marrow, ooh, for people who have worked with bone marrow, it is difficult in the best of circumstances. But once again, it was very encouraging. And then human prostate. Oh, beautiful tissue, solid tumor. It's human. It was like your best case scenario to figure out your proof of concept. And sure enough, everything worked great. We did have a couple of markers that just didn't work, which is kind of expected because the tissue had been through a lot. And then we had a lot of the markers were dimmer. And we were able to compare this to a non-xenium phenocycler only, because we had additional sections so that we could do that. We had the positive control, right? What it should look like. We were able to compare the xenium to it. So now we have this cool workflow. It is obviously a group effort. The Phil runs the xenium, walks the slide down the street. We're all on the same campus, but we are buildings away down the street to our lab. We run the phenocycler. We put it through that process to take the flow cell off, which is really adhered to the slide. So it's quite a process. You get it off, and then you could take it downstairs to experimental histopathology for the staining for H&E and doing the scanning. So now that we have everything all set, we could do our first legit non-proof of concept project. We are still working out which markers are definitely not going to make it, but at the very least, we are confident that we can move forward. The Phil had a new project. This one it's still using the Xenium V1. So you're looking at hundreds of targets, and they wanted to build out a custom IO gene set to add in. And so they got 4 different investigators, so lung, prostate, the one that you saw earlier, a couple of others, and said, hey, we will run Xenium for free. It's like the second project where we got free Xenium out of it. We really love the Phil. Anyway, so we get this free Xenium and then said, hey, we'll run the phenocycler, which was kind of no choice because this is my boss's. So of course we were going to do this, but my core is this division hybrid. I never fit well on an organizational chart. So we do a lot of collaboration as part of our division world, but then we're also fee for service as part of our shared resources core world. So this is our division boss. And we had 4 samples where we had run through Xenium, and then we had the next sections, apparently not serial sections, as you'll see in a minute, but within a couple sections. And we ran a 46-plex. 22 of the markers were in both the gene set and the proteins. Once again, we had good morphology. Most of the markers were comparable. A lot of them were dimmer. There were only a couple that didn't work. The H&E, in general, we still see this trend of the H&E from the Xenium phenocycler as expected, slightly less crisp, but still pretty good. The top one is the post-Xenium. So we do scan these at 40x, which the pathologists tend to prefer anyway, but this also helps mitigate that less crisp edge so that you can see things a bit better. Now we have this little other question that we've been working on that we still want to nail down is during the Xenium process, they add a quenching step. And this quenching step is kind of purple. So you need to remove the quencher or dequench it before you do the H&E, because otherwise your H&E tends to be a little dark and the pathologist can't read anything and people get upset. So now the question is, do you take off that quencher before or after you do the phenocycler? They say before you're going to do anything with IF, leave it there, take off the quencher afterwards. Long story short, we had been removing it beforehand. We had very little background, had that little bit of dimmer. So I'm actually concerned that if the quencher was still there, it might even be dimmer than that. But we hadn't tested various combinations. We just had been running it. Everything was great. We had another project after this that after conversations with 10x and with 𝐒𝐤𝐿, then we went ahead and removed the quencher after the phenocycler. We had a couple of other mitigating circumstances where this was running the Xenium Prime, which sounds fancy, but it's the 5K. So it's on the machine for a couple of days longer. And they also did the cell segmentation markers. And needless to say, a lot of our markers were dim, much dimmer than what we saw before, but now we don't know why. So that's one of the things that we want to work on. But getting back into the differences, so a little bit of eye candy. So you have the multiple colors, but then we'll get into some of the markers. Excuse me. So the top row has the post-xenium phenocycler. The bottom row is phenocycler only. And here you can really see the difference between the morphology of the two. These are obviously not true serial sections. They're a couple sections apart, but you still get the gist of it. The pan-keratin, same sensitivity, specificity, intensity. Worked great. In fact, we found that all of the keratins performed great in both. The CD8 looks dimmer, but it's actually still as sensitive and specific. We just would need to use a longer exposure time. We did everything kind of the same, so you have the good comparison. We just cranked up the exposure, it would look more comparable. If for some of these that are dimmer, if we just increase the concentration a little bit more, that's also going to fix it. And then we have CD163, that poor macrophage. The other macrophage, CD68, looked great. CD163 did not, but it's almost like a hint, like it wants to be there. So is this just a matter of needing to increase the concentration? Hopefully. This might also be a case of when you are putting all the antibodies onto the tissue, and then you could do that kind of in batch, one big master mix to do all of that. But when you go to run them, then you're having a fresh reporter plate or your plate of floors for each run. And each of these can only be run one at a time on our system. So that could have even been a technical error. So that's one other thing that we need to figure out is, does CD163 after survive, because we kind of need it for our project. But one of the other things that's important to note for anybody who cares about neutrophils, aside from my boss, is that neutrophils are really hard to find in the transcript world. They're just not really there. So you need the proteins, but they're also delicate creatures and you tend to lose them pretty quickly. The neutrophil markers work just fine. Now we're going to get into some of the stacking or the overlay of the proteins and the transcripts. We used Xenium Explorer, like a lot of people have recommended, which is great if you have your tissue has not moved at all from going from one assay to the next. And I know, spoiler alert, it's going to shift. You're going to have that slight shift, and Xenium Explorer is not set up for that. So you need something that's going to be able to warp. Most alignment software does kind of warp one so that you have the exact fit. This one doesn't. So we had to choose, do we want everything to be aligned up at the top of the tissue or the bottom of the tissue? So we're in this little area here that everything is aligned. We are working on the whole issue of alignment, but at least temporarily, proof of concept. This part looks great. Awesome. So here we're looking at keratin in white, CD8 is in red, and over on the far right, I also included DAPI nuclear stain for a bit more context. Now we're looking at some of that overlay. So from within Xenium Explorer, over on the left, we have now put transcripts. So the keratin transcripts in green and the CD8 transcripts in red, and you see that you have this beautiful alignment. It is interesting, though, to see that there was a lot of these keratin transcripts background, so to speak, kind of scattered throughout the tissue. And they were like, yeah, that's part of the background. I'm like, cool, you guys have the same problem that we do with proteomics, only ours is uglier. So we have next is the cell segmentation. So taking the clusters, which is why there's a bunch of different colors, but taking the clusters and just superimposing them onto the proteins. So this is the Xenium Explorer segmentation from that part, from the Xenium part, and now we're overlaying it onto the protein. And we see that it looks pretty good. There's a couple cells that might be missing, but otherwise it might be okay. So maybe for now, since we can't get that perfect alignment and that's still being worked out, maybe we could at least get some preliminary data by analyzing them separately and kind of combining data on the back end, right? So use Xenium Explorer and get your Xenium data, and then use QPath is kind of our favorite cell segmentation viewer, just kind of general, easy to use, and it's free to take care of the PhenoCycler. One problem. Over on the right, QPath does a great job with cell segmentation. Nothing is ever perfect, but that's pretty darn close. The script that we use, we actually got from one of 𝐓𝐢𝐭𝐥𝐞's people, and it's very similar to CellPose or DeepCell. These are really good scripts. On the left, you may notice that Xenium Explorer, not quite as good. And very importantly, look at the number difference. We're talking 20% difference. So there is no way that we can analyze them separately and then be able to put that data together on the back end and say, we think that this is cell-to-cell data, because it's obviously not. So we do need to get this alignment figured out. Best case scenario is you align the images using some sort of, shape shifting thing. So they do align, and then be able to do cell segmentation on one of the images, take that mask, the segmentation mask, put it onto the other image, and now you can have that cell object data so that each cell you have its XY coordinates, you have its intensity values for each of the proteins, for each of the markers, and you have all the transcripts. So, and then it's diving into the data would be the next step. Now, there is a company, there are a couple of companies that have been working on it. There's one, I think it's Aspect Analytics, that appears to have that workflow pretty well figured out. But once again, I will say the word budget, and you will know why we're not doing that. So the next step is going to be diving into that data. One thing of note, muy importante, is clustering those proteins, right? So you segment the cells, you hand them off to the bioinformatics person, they do the clustering, they're like, oh my gosh, look at the numbers, they're great. But if you take those clusters and you put them back over the protein and they don't align, then something is wrong. But how often does the bioinformatics person take the cluster and put it over the protein? Because I tell you what, about half the time it doesn't align. That heat map for your cluster says this is where all of your CD8s are, and then some of your samples, when you have multiple samples put together, then some of those samples is, there's no CD8s in the cluster when you look at the protein image. So there is some disconnect. It just means that things need to be reclustered, but that is one of the ways to QC, is to actually put it over your image. Next steps, we are continuing to optimize the workflow, always open to collaborations, dealing with the analysis, data viewing, sharing, and storage is its own thing, which a lot of y'all are understand, but even within the hutch, it's difficult, which is wonky. And so a big thank you to obviously my boss, 𝐒𝐤𝐞𝐚, but Kristen Robinson is the one that's in the trenches, actually running the PhenoCycler. Xiaodong Zhu is part of 𝐒𝐤𝐿's research lab, and he's the one that has also done a lot of orthogonal data on the samples that we're using. Anna Els in the Fred Hutch Innovation Lab is the one that ran the Xenium. The investigators that I've listed are all the ones that provided us samples, and then Zhulan Yang is helping us with dealing with Xenium Explorer. We're part of a Codex user group, and Diane Bender was the one that gave us the protocol for the to take off the flow cells. And then, of course, 𝐓𝐢𝐭𝐥𝐞 has been very supportive. In fact, they're the ones that first told me about the JAX protocol. I'm not going to go over all of this, but just the fact that even with just proteomics, it takes a village. We're working with pathologists. Our sister CLIA lab, the whole technology team, the investigators, the bioinformatics. This is a group effort. So any questions?